Good afternoon, everyone. I really can't tell you how happy it makes me to be here. I am here today with the United States Embassy as a scientist with the US federal government, but I was telling some of your colleagues and senior leadership here, this is my first trip to Sri Lanka, and when I look around me, it almost seems like I'm back in a part of the world which is very close to where I grew up, and people are assuming I'm a local, and I'm almost feeling ashamed that I don't speak much Sinhalese. So, in a sense, it almost feels like coming back to a part of the world where my early career had started, and that makes me very happy. So I'm here mainly as a storyteller. I hope to tell you a story about genomics and how it affects all of your lives, and if you're a doctor, how it is changing the way medicine is practiced. Because there's quite a bit in the talk, I'm probably going to try to give you, before we get too deep in, just a sense of what's in here. I want to relate to you as an audience. I am, like many of you, a fellow academic, now a federal employee. So just a little bit about me and where I come from and what brings me here. We will then move on to just learning about genomics. Many of you are students of medicine or biochemistry and fields where you might have heard of genomics. I want to give you a little bit of a 360-degree view of what is genomics before we get too much into genomic medicine. And part of that is the story of the Human Genome Project, which is a very fascinating story. And then finally, we'll talk a little bit about how genomics is fundamentally changing the way medicine is practiced. Because I do believe that in 10 or 15 or 20 years, doctors will use DNA as a diagnostic tool or therapeutic tool, just like they use their stethoscopes today. So this is a little bit of the schematic of the talk. Most importantly, for me, these talks become that much more fun when I get questions, okay? So during or after or any point while I'm here for the full day, please let me know. No question is a silly question. Just ask me anything that's on your mind, and that's the way that I get to engage with you as an audience, okay? So that's my take-home message at the beginning. I am very fond of questions. Okay, so I am here today as a guest of the United States Embassy in Colombo, but my affiliation is with the US National Institutes of Health, NIH. Some of you may have heard the acronym, some of you may know the name. Mostly when you think of Washington, DC, you think of the White House on the left or the US Capitol. Of course, both of those have been in the news in the last week, and we don't get as many tourists as either of them. But my agency is that large brick building on the right of the slide, actually one of the biggest brick buildings in the world, a teaching hospital just like this one, a research hospital. But NIH as a whole is the single largest funder of biomedical research in the world. Even under economic tight conditions or even under times of hardship, we managed to put more money into scientific research than any other country anywhere. That includes Russia, China, the European Union, and especially during the pandemic, NIH had a global role in making sure that the vaccine was manufactured and sent out, not just within the US, but globally. So this is my little corner of the US healthcare system, and I'm here to bring to you all of the wonderful things that NIH is doing in the field of genomics. What I do at NIH can sometimes confuse people a little bit, so I want to give you a sense of what my job is. I am a former scientist. For 10 years or so, I was what you would call your classical academic faculty member, running a lab, doing experiments. About 5 years ago, I took this job, which is taking taxpayer money that NIH gets from the US government and learning which kinds of science need more investment to do better, or what is a very new thing just starting to happen, where a scientist or a group of scientists really needs financial support so that their science can progress better. So this new aspect of my work for the last 5 years or so is a little bit more like an investment banker. I take taxpayer dollars and I find good scientific investments for those dollars. And then the third aspect of what I do is administration. Once we give out that money, which is sometimes tens of millions, even hundreds of millions of dollars, we have to make sure that the money is getting used and people are publishing good research, training young doctors, training young people in the lab. So there's a third part of me, which is more like a science administrator alongside the former scientist or the investment banker. So this is just to give you a sense of what it is that I do and how it relates to things that I want to bring to you for the rest of this talk today. I was born, as I said, I was born in the UK, but I moved back to India very early. My undergraduate and master's degrees were both in the University of Calcutta, not too far from here, which at the time was what I would call a fairly resource-limited institution. In 2004, I moved to the US to get a PhD. So in white is the Indian part of my career, and in yellow is the rest of my career after that, which was I got a PhD. For a few years, I studied gut microbiomes, which is another very, very fascinating area of genomics. And then finally, for 5 years or so, I've been doing that job that I talked about. So this is me in a personal sense. The question could be asked, I just wanted to also add some pictures. That's me as a kid in Scotland before I moved back. For anyone from Kandy or anyone that lived in a tea garden, on the top right is a picture of an Indian tea garden, an old house in an Indian tea garden in Assam with my parents. And then this is me just before I moved to the US and just after I came back in time to marry my wife before she married someone else and I lost her forever. So this is, a little picture of me as a person and a scientist, so that I don't seem like someone that is far away and talking about abstract concepts such as DNA sequencing. Now, what brings me to Sri Lanka? Of course, I grew up watching your cricket team. For many of you that were born in 1996, and some of you are obviously not even born at that point, I stayed up all night watching every ball of that game. And of course, I cheered for Sri Lanka. We didn't cheer for Australia. So I have always been curious and I wanted to come here. But then the weather in DC, as you know, is like this thing now, except it's not so clean. That looks like nice and clean white snow. It's actually muddy, horrible, slushy snow. So when the US Embassy put up this fellowship program saying you can spend a month in Sri Lanka and this is what the beach on Golfest Green looks like, I was like, okay, I'm going. I just have to go. So, but jokes apart, I'm here because I want to bring a training program. I want to work with doctors and nurses and computer scientists and laboratory people in Sri Lanka, helping this country to have the computer science ability to practice genomic medicine. I'll talk more about the actual theory of it. But most of the reason I'm here is helping Sri Lankan healthcare professionals, not just doctors, as I said, but others working in allied healthcare professions, to become able to practice genomic medicine in their patients, in their hospital systems. So this is overall, I hope, a little bit of context for what brings me here. With this, let's actually get a little more into genomics and why should you care? What is genomics? So I like to use a philosopher's approach. Monks or philosophers teach us to look into our mind, understanding the basics of our mind and our soul. On the other hand, in our physical aspect, in the aspect of our bodies and our health, until we understand DNA, we cannot really understand what is in our bodies. We are building blocks coded for by DNA molecules. And so genomics is basically the science of understanding your own DNA and what are the secrets to your health or your heredity or your disease risk. All of these things are encoded in this molecule called DNA, which I'll tell you more about. And genomics is nothing but the reading of this code and understanding what makes us who we are in a physical sense. This is increasingly relevant in daily life. In the US, people do it as an ancestry. A child will ask its parents, "Mommy, where were my great-grandparents from?" And at that point, DNA can tell you if they're from a Native American tribe or if they're from Europe. So this is not just a healthcare tool, also a way of understanding your ancestry. This is a career field where so many jobs of so many types are coming out of genomics, that especially for people at the beginning of their careers, I often say genomics is not just science, it's also a future profession for you. I'm going to tell you in the next few slides more about the basics of genomics, but I want you to know that whether or not you realize it today, genomics affects every aspect of your lives. Let's do some basics. History of biology. I think 200 years ago, all that we knew about living organisms was that they were made of cells. Microscopes were not yet evolved to the point of looking inside cells and understanding structures. Maybe 150 years ago, microscopy got to the point where scientists realized that within a cell is something called the nucleus, and the secrets of life or heredity get passed on from one generation to the other through these smaller pieces in a cell called a nucleus. Okay. Another 50 years, maybe 1890s or 1900 or so, finally people realized that within the nucleus there are chromosomes. And it's not the nucleus itself, but what gets passed on from mother to child or father to child is the chromosomes. Okay. What people still were wondering is, what is the magic molecule? We can see these chromosomes passing on, but we still need to understand what is the real chemical entity inside a chromosome that helps pass information on to succeeding generations. I'm going to give you the short version of the story, but in the year 1953, Watson and Crick discovered that it's DNA. It's this molecule based off a sugar phosphate backbone with this lovely double helical structure. Think of it as a chemical molecule which has two strands of a sugar phosphate string with 4 nucleotides, A's, T's, G's, and C's. So there are 4 basic building blocks of DNA, the 4 nucleotides. And these are sort of strung together like beads on a necklace. And then the necklace is twisted on itself to form this double helical structure. This is the molecule that codes for all life. Okay, so this understanding that DNA is what was actually passing information through generations began in some ways the study of genomics. We now understood what the molecule was. We still needed to understand how it works, but at least in the 1950s, we knew it was DNA. So I want to give you a sense of what DNA looks like and how much of it you have, because this is a humongous amount of information within each person. So human beings have 46 chromosomes. You start stretching out the DNA in those chromosomes, it basically starts looking like a long series of those 4 letters, A, T's, G's, and C's, okay? Now, what is staggering is that one cell, just one cell, if you take all of the DNA in one cell, it has 3 billion letters, 3 billion DNA bases. That's 300,000 letters of DNA in just one human cell. What gets interesting? Each of us has about 30 to 40 trillion cells, okay? I'm just trying to give you a sense of the amount of DNA. In one cell, 3 billion base pairs. If you take the DNA and you start stretching it out as a string, it would be 6 feet in one cell. If you take all of the cells in your body and you take all of that DNA and you now start making a string out of it, that string would go to the moon and back 1,500 times. Each of us contains that much DNA and that much information, and somewhere within all those letters, all those A's, T's, G's, and C's is hidden the unique secrets of who we are as unique, wonderful human beings. So DNA is the code of life, and more specifically, the code for who you are as a person. Another way of looking at this is just as the scale of a book. Using this funny analogy, if you type out the 3 billion letters of DNA in a standard book-sized format, that book would have 1.2 million pages. This is also where I start saying that computer science and DNA get very closely linked to each other. If we have to understand your book, your DNA, no one of us, no scientist, not even a Nobel Prize winner can read 1.2 million pages and understand all of it. This is also a point where I want you to start thinking about why computers are so important to making sense of genomic data. Okay, so that's, I told you that each of us contains a lot of DNA, but why does it really matter? Why it matters is that essentially within those strings, within those long series of A's and T's and G's and C's, almost every single physical trait you can think of, your height, your eye color, skin color, your risk of cardiovascular disease, if you have anxiety, maybe a predisposition to anxiety, if you ever develop cancer, which cancer medication will work best for you, how long you will live, will you get Alzheimer's? All of these qualities are already encoded in your DNA, and it's only through understanding all of those base pairs that doctors can start approaching you as a patient and saying your DNA is giving me a clue to treating who you are. Think of it as the sum total of your physical existence in a hidden code, and genomics being the field of science that will help you understand that code and help doctors treat you if anything ever happens to you. I do want to say that genomics is not everything. Your environment, your social exposures, behaviors you learned at home or in school, these also count towards making you who you are. But from the physical aspect, it's almost 100% a DNA and influence in making us who we are. So you have environmental exposures, you have physical exposures, and DNA together with the environment is making us, each of us, unique people, and we need to study it from that aspect. Okay, I talked about the genome. The genome is the sum total of these bases, all of those 3 billion letters. What are genes? Many of you, if not most of you, have heard the word genes, right? So in that long string of many, many base pairs, only a tiny fraction, 2% of the 3 billion base pairs actually create a protein. If you take the code and if you decode it, it goes from being DNA into something that is getting built into a protein. So that tiny fraction, that 2% of the genome, is what we call the exome. And it's the tiny piece that codes for an actual protein. Roughly speaking, every one of you has 20,000 genes. So 20,000 genes, 20,000 proteins. As a coincidence, most modern cars, if you average out across manufacturers, a modern car has about 20,000 parts. A Mercedes a little more, Toyota a little less. But mostly a car has 20,000 parts. The human body has 20,000 genes. Now, as biomedicine students or as doctors, that the human body is a much, much more complex machine than even a very expensive car, right? So where genomics becomes really important is saying, how do these genes work together, and what can we understand from studying the ways in which 20,000 genes interact in health and in disease? And that is the biggest mystery that stands before us. Maybe 20, 25 years ago, this was a mystery, but we did not have the tools to understand it. Just pure molecular biology had not progressed to the point where we could make any real experiments helping us understand how two genes are interacting or what happens when a gene is deleted or another gene is inserted. Some of you have heard of these progressing tools. DNA cloning is basically a set of methods where instead of having to work with massive pieces of DNA, scientists could cut out small parts and grow them in cell cultures. Think of it as a tool where you can get scissors to study small pieces of DNA, which is important because 3 billion is too big to study, right? DNA sequencing is basically machines that will help you read DNA. As I mentioned, DNA is A, T's, G's, and C's. And until about 1977, we had no machines where we could read the order of these bases. We had DNA, but we could not read the code. So DNA sequencing was the second big progress in that state. Polymerase chain reaction, PCR, that was another Nobel Prize winning effort. DNA in a cell, you have a very tiny amount of DNA coming out of any one cell. It's long, but in terms of actual mass, it's tiny. PCR was a way where we were suddenly able to amplify even a tiny portion of DNA and get enough that we could now do other experiments. I'm just going to shout louder so we can keep going through this. Okay, so the screen I see is up here. So bear with me and just tell me if you're having a hard time listening to me in the back. Until the power comes back, I'm just going up on my body. So PCR then was the technique where we could take DNA and grow enough of it that scientists could do more to understand the genome. So at this point, I want to talk about the shift from genetics towards genomics. So having learned about what is the genome, what are genes, the next big revolution was being able to study all of the genome rather than one gene at a time. In 1907, when we still didn't know about DNA as the molecule, scientists took things like, I take a plant with yellow flowers and I take a plant with green flowers, and I study the color of the flowers as one gene. I'm going to mate the two plants and see if the flowers are both yellow and green or only yellow, only green. Basically, scientists at this point were still studying one trait, one condition, one gene. DNA The structure in 1953 made it much more clear that we have many genes, tens of thousands. And in 1987, scientists finally said, we really need to move beyond studying one gene at a time, because obviously if we have 20,000 genes, we need to study all of them together to understand how they interact and how this complex machine of the human body is working as a sum total of all of its genes. So this is the transition from the older field of genetics, which is still there and is still very important, to the newer field of genomics, which by definition is the study of all of the DNA at the same time. One of the things that happened around that time is scientists said, if we want to study genomics, we have to be extremely brave, extremely audacious, and say, can we sequence the full human genome? We now know that humans have 3 billion bases, and we know we have 200,000 or so genes, but just running sequencers so that the full genome of a person has been completed was considered almost impossible, and people were sometimes comparing it to how much work it took to put a human being on the moon in the year 1985. It was just extremely challenging to even think about sequencing a full human genome. Why is that? Well, number one, as I said, the genome itself is very long, 3 billion base pairs. Number two, the machines in 1977 or 1980 or even 1985, a machine could only sequence 500 base pairs at a time, maybe 800 if you had a really good one, okay? So you take 3 billion and you take a machine that can only sequence 500 or 800, and you can see it was tremendously logistically challenging to even try doing that technology and getting a full genome. Even worse or even harder, you don't just sequence a piece of DNA once and done with it. DNA actually has to be sequenced many times over the same molecule before you can get a high confidence sequence out of it. So it would have been 3 billion times about 30 in terms of the actual number of sequencing that needed to be done. And finally, much of our genome, maybe about half of the genome in a human, is pieces of DNA that look really similar to each other, things that are just repeated over and over and over and over. And so what happens is when you sequence something like that, you have a really hard time saying, where does this piece fit? Is it from chromosome 17? Or I know there's another place on chromosome 6, which is extremely similar in sequence. So it's just really challenging to take sequencing data and use computers to map it back to the place that it came from. So all of these are factors that sort of underlie why the Human Genome Project, when it set out, was viewed by many as almost doomed to failure, or at least very impossible and not going to be completed in anything less than 40 years. But I like to say that this is where scientists who think big sometimes get ahead in the game. In the next few slides, I'm going to tell you why this succeeded and how it succeeded, and maybe lay the groundwork for where genomics is going from that point. So I think the first thing that happened is that scientists across the world, this is also an example of international collaboration and scientists coming together and saying, let's do this as a team. Because 3 billion nucleotides, even one country in the 1980s, even the US, all of the sequencing centers in the US together could not have sequenced that much DNA. So the first thing that happened was countries and labs divided up. The bigger chromosomes, 1 and 2, were left in countries that had more sequencing capability. Some of the smaller chromosomes were sequenced in countries that were already in genomics, but maybe had a little few sequencers. There was an international agreement and collaboration into dividing and conquering this massive genome project instead of one country trying to get all of the credit for it. My postdoctoral advisor, Eric Green, was the person who sequenced chromosome 7 at Washington University at St. Louis. Others in Boston sequenced chromosome 1. But it was an early example of global team science, where to attack this massive biological problem, scientists really were able to create what you would call mini United Nations for genomics. The other thing that was done was to break up the genome. As I mentioned, we had the tools such as cloning that allowed genomes to be manipulated. The whole genome is 3,000 megabases. A chromosome, average chromosome is about 130. But we were able to use those tools to cut into smaller clones. The larger ones were about 1 MB, the smaller ones were about 0.2 MB. And that size is the size at which a lab can say, This much DNA I can handle. This much DNA I can sequence and make some progress. So it was essentially a question of taking a massive, challenging project, distributing it up between countries, and then whoever got a piece was also able to use molecular biology to cut their assigned chromosome into smaller pieces that were actually sequenceable and manageable from a laboratory point of view. The other big advancement is something called shotgun sequencing. As I said, the sequencer back then could only sequence 500 or 800 nucleotides of DNA. This really interesting approach came up of, instead of trying to take this very long string and start sequencing from one end and going from one end to the other end, which would have been almost impossible, let's do this thing of taking the DNA, using enzymes to cut it up into many smaller pieces. Those are the subclones that you see here. Take all of these small pieces and sequence them. So each piece is now going to fit within a sequencer. And once again, the wonderful collaboration of computer scientists came into the picture. The computer scientists said, sure, you do that, and then you give us back these smaller pieces of DNA, and we will write the computer programs that will help connect them to each other. Because we know if we take one piece and it has a sequence at the end, and we take another piece that has the same sequence at the beginning, then by logic rules, those two pieces sort of link up to each other. So this was the early confluence of computer scientists and genomic scientists getting together, where genomic scientists did the first part of breaking DNA up and sequencing it in small pieces, and computer scientists did the second part of taking these small pieces and reconnecting them back into full-length genome sequences. This all became a sort of race. I was still too young. I was not yet in grad school, but Eric and others tell me that at one point there was a significant intellectual battle about when the human genome was sequenced, whether it should be viewed as a for-profit exercise, and a company called Celera Genomics said that we will sequence the human genome first, and we will go to the Supreme Court and we will take out intellectual property rights on the genome. And after that, if someone wants to use the genome for drug discovery or to treat cancer, then we will sell licenses to different parts of the genome. So they had really good technology and they worked very, very hard, but their approach was clear. The genome to us is no different from a new drug trial. If we sequence the genome, we will sell access to the genome. NIH had a fundamentally different approach. NIH's approach was the genome belongs to all humans. We may be a little slower because we have to work across countries, and we will do this as a group, and we will release all our information. But if we sequence the genome, then we will not try making anything of a commercial product out of it. I'm going to simplify things somewhat to say that these two groups were racing each other and with technologies and people. And at the end, the White House had to get involved and say that we cannot do this on two separate fronts. So President Clinton came into the picture and sort of brokered a truce. I will also say that this was not a fair race. The company, Celera, that I mentioned, to do the sequencing their way, they actually were using all of the information from the NIH Public Human Genome Project. The company depended on having access to the public genome data, because without it, their computers could not assemble. On the other hand, the NIH group was doing the exact opposite, where every night they were making all of their data available. Anyone, any scientist across the world could get access to their data overnight. But they were not dependent in any way on the Celera genome. So this was two groups trying to do the same thing with fundamentally different moral or ethical approaches, and one of them being dependent on the other. But it was a race, and to the extent that both groups knew that the other group was working hard, in many ways this led the Human Genome Project to progress at extremely rapid speed of growth. The press got into the picture, journalists like framing everything as a battle. The two people you see here on the front is Craig Venter, behind him is Francis Collins, both colorful personalities. And for months or years, the general public was super, super excited about genomics, because who's going to finish first? Who is going to finish first? I'm happy to say that the race effectively ended by being declared a tie. The president and others negotiated between those groups. In 2001, almost in the same week, the Celera genome was published in the journal Science. The public human genome was published in the journal Nature. Both groups came together and did a joint press release. in a sense, the science one. And of course, the Supreme Court came in and said that we cannot allow the genome to be sold or patented in any way. So the ultimate purpose of NIH, which was to keep all of this genome information in the hands of any scientist, that was also accomplished. The genome was sequenced, and it was free and public for anyone to use. About 2 years later, so this is all being done in a rush. At this point, they published the genome, but it was still a little bit short of what you would call a polished product, because of course everyone had been working day and night just to get it out there. About 2 years later, they filled in all of the gaps. They sort of polished it up and made it more usable for even the most advanced purposes. And this is just before I was entering my PhD program, but this is officially now considered, we have done it. The Human Genome Project is over. In 1985, we were not sure we could do it, and many people thought it was a waste of money. But this was the point at which the human genome was no longer a blockade in understanding how those 20,000 genes work together. It was considered done. US Congress established National DNA Day, which we celebrated HGRI in a big way. And sort of the stage was set for the next part of genomics. I also want to make this more relatable. I should not talk about these topics as if they were happening only in the US or only in the UK or Western countries. Around that same time, Professor Vajira Dissanayake and others, collaborators both within Sri Lanka, close by in India, the complete Sri Lankan genome was also sequenced and published just a couple of years after the initial Human Genome Project. And they also used a computer science approach to map where in the genome are Sri Lankans unique relative to other populations. And they published what is called the Sri Lankan Genome Variation Database. So genomics was not happening just in the Western world. Genomics was also happening here. And these data are still out there and public for anyone to use. Where do we go from here? So in 2003 or 5 or 6, we knew the genome was done. So you see those two scientists celebrating, congratulating each other because they feel like they had a big puzzle and all of the pieces of the puzzle have now been filled in. So they feel like their work is over, right? They're in a happy frame of mind. I think soon after, within at least 1 or 2 years after, what became really evident was that sequencing the genome was actually the simple part. It sounded tough and it took 15 years and it was very expensive. But what we had very little idea about what the biology was, how the genome works. So instead of that small puzzle and 2 scientists, think of a puzzle with 3 billion pieces. So you see the other group on the right. They say, okay, we sequence the genome and now we have a puzzle that is 1,000 times more complex. And we haven't even made any progress in understanding the ways in which genes interact with each other. This was the end of one race and the beginning of another much harder race. And in some ways, that race still continues. What I'm going to sort of move topics and talk about for the last 1/3 is genomic medicine. So while scientists try to understand the very, very complex biology of the full genome, do doctors have to wait before scientists say, we got it, we now understand everything about the genome, now you can do genomic medicine? The answer to that question is no. Doctors, even today, are actually able to take genomes and use them as tools in their clinical practice. This is a new field called genomic medicine, sometimes called personalized medicine, individualized medicine, precision medicine. But basic premise of this is that for each patient, what we already know about the genome is a good way for a doctor to say, I want to start with this drug for this patient, or I want to start with looking at this panel of genes for that syndrome, because I know that the genome is already a diagnostic tool and a therapeutic tool. And while the basic scientists take another 10 or 15 years to study basic science questions, I already have enough genomic knowledge to start using the genome as a clinical tool. So with this, I'm going to pivot towards the more, what you would call the hospitalized use of genomics in US healthcare systems to treat patients quicker and better. So how does this happen? Of course, you have a patient that comes in, you sequence their genome. I'm happy to say that at this point, the cost of sequencing a genome is 350 to 500 US dollars. That first genome was 3 billion US dollars. Right now, even the average middle class US family the cost of genome sequencing is something that is not prohibitive, and insurance companies are starting to cover genome sequencing on a routine basis. So a patient comes in, you get the patient's genome, you get a family member that does not have the same syndrome, or you have a sibling that does not have the same rare genetic disease, you sequence another genome. Computer scientists come into the picture, they compare the two genomes, and they identify the exact places where the person with the disease is different from the person without. They give you back what is called a list of variants. The doctor suddenly has a small number of places at which the patient has genomic changes that are unique to them, and that is where genomic medicine starts to happen. As with the genome itself, I do want to leave you with the fact that genomic medicine is already happening in Sri Lanka. Before, in about 5 or 8 years ago, genomics in Sri Lanka was being used as a tool for understanding historical populations. That's the paper up on the top left. But especially in the last 5 years, 7 years, I'm seeing more and more and more papers coming out every week where doctors and clinicians in Sri Lanka have prescribed genetic tests. The bioinformatics has been done often at the University of Colombo or by someone that has decided to outsource it to Singapore or India. And then a patient has benefited from the application of genomic medicine in Sri Lankan hospital systems. The most recent example that comes to mind is a baby at the Anuradhapura Teaching Hospital about 2 weeks ago who was treated for congenital methemoglobinemia and is now doing really well with high doses of ascorbic acid prescribed based upon the genomic finding that the doctor found for her particular cases. So this is not an abstract NIH talk. You should be already able to access the expertise within this country if you plan on using genomic approaches in your medicine. Okay, let's go through a few examples. Where is this happening? What sorts of areas within disease? I'm going to start with cancer. Most people do not think of cancer as a genomic disease. I want you to know that the very basis of all cancers is that the DNA was messed up. The DNA was mutated beyond a point. One cell accumulated a few mutations on the left. Maybe this is a smoker and they just kept smoking and kept smoking and kept smoking. And then instead of 1 cell, it was 2 cells with mutations, 2 to 4, 4 to 8. And what happens when these cells keep accumulating a mutation load is at some point the cell cycle, the rate of growth of cells, goes completely wild. And the cells go into a growth rate that is far, far more than what their normal biological growth rate would be. And that abnormal growth of cells is essentially what is a cancerous tumor. The beginning of a cancerous tumor, once again, is a genomic change. So cancer, more than many other diseases, the predominant disease worldwide that is a genomic disease is cancer, all cancers. One of the most prominent ways that genomics and cancer intersect is in diagnostics, finding what has gone into a cancerous state. Historically, pathology, a patient came in, they presented when they were already having symptoms. Doctor said, I think I see a tumor on your x-ray, or I think I see something where we need to do surgery. Surgery would happen, a sample of the tumor would go for a histopathology test, and then it was getting diagnosed as, I'm sorry to report, but you have cancer, okay? So this is usually happening after the disease develops. Plus, all you can tell is that the tumor was cancerous or precancerous. Genome sequencing of cancers is more like a microscope that lets you tell exactly what happened with that person and that tumor. Because now you can say, instead of just saying, I see on a slide that your liver has a cancerous portion, you can say, your liver DNA is showing me changes in genes A, B, and C. You have cancer, but at least I know which genes have been mutated, and let me go see if a cancer drug would be particularly useful given your liver cancer. So genomics is making it possible to treat cancer with a much, much more precision approach relative to older approaches. The most common way of doing this is a liquid biopsy. As I mentioned previously, biopsy was happening after surgery or at suspected disease. Now, I just turned 45. I could go in, I could give a small sample of my blood, And if there are any cancer cells anywhere in my body, chances are that the tiny amounts of cancer DNA just coming out of a cancer cell into my bloodstream can be picked up with a sequencer. And a doctor can do a routine. I just did this for colon cancer weeks before I came here. But screening for cancer is now completely possible with genomics-based approaches. No surgery needed, no fine needle aspiration needed, and people can be screened on a much more routine basis for the genomic changes that would indicate that they have cancer of any sort. So this is really changing not just the treatment, but also the initial detection of cancer. And we know that cancer, the quicker you find it, the better the chance you have of treating it successfully. I'm going to pivot and talk about newborns. I don't know if this hospital system also includes the neonatal unit, but genomics is probably the single biggest change in genomics has come from treating babies or children for whom previously a diagnosis would have been impossible to find. One of the biggest ways there is non-invasive prenatal testing. Before genomics, if there was a consanguineous marriage or there was reason to believe that the fetus had an abnormality, the only way of actually testing for it was to do amniocentesis, a fairly invasive procedure where the expecting mother comes in, a fairly large needle is inserted, and a portion of the amniotic fluid is drawn out. That gets isolated cells from that, karyotype is done. It was not something that would be done unless there was a reason to expect that the baby was at risk. And even then, it was still a pretty invasive procedure. Just as with liquid biopsies for cancer, now almost any family where any sort of chance of genetic diseases is even remotely possible, the mother comes in, we take a small sample of blood, because the fetus releases tiny bits of DNA from its cells into the blood, and genomics techniques have become capable of sorting out the mother's DNA from the fetal DNA. We can sequence the DNA of the fetus long before delivery, and we can almost always see if there's anything of concern that should get started, treatment should get started even before the delivery happens. So this is fundamentally changing neonatal medicine. I think the #1 test worldwide among neonatal medicine practitioners is now genomic testing of the fetus before it's born. This is making it possible to treat things that before would just have been, we don't know what's wrong, let's keep our fingers crossed and let's see how the delivery goes. Just giving you some numbers, this is from the NIH neonatal ICU. 44 cases came in a certain window a couple of years ago. All of these are diseases of unknown etiology. Mostly the physician would unfortunately have had to tell the parents, we know your child is very ill, but we don't know what is going wrong. We are going to treat it symptomatically and we are going to hope for the best, but we don't have any real diagnosis for what went wrong with your baby. That was the older way of practicing neonatal medicine. Once you get genomics into the picture, 37% of these children, actually they found something wrong within the genome. 26% of those, they were able to say, we don't just know which gene is messed up, we also have a treatment. We know a drug, an intervention that works in this gene. And consequently, just the cost of treatment alone went down by almost $15,000. But more importantly, these children in the pre-genomic era would just not have had a good shot at getting any sort of treatment. They would have been treated palliatively, symptomatically, and given the success rates for extremely sick newborns, it's almost unfortunate, but fair to say that most of them would not have lived out to adulthood. So genomics is fundamentally changing the practice of neonatal medicine by making it possible to find a diagnosis for neonatal cases which before would just have been given up, and now we no longer need to do that. Okay, I'm going to finally start wrapping up with pharmacogenomics. We talked about cancer, we talked about neonatal medicine. Let's talk about pharmaceuticals and getting the right drug to the right patient. You may know that right now, the way clinical trials operate is essentially on a statistical hit or miss basis. You enroll 100,000 people, you split them into 4 groups, you treat them with a drug. 3 of the 4 groups are not going to get any benefit from the drug. Maybe the drug worked but was too toxic. Maybe the drug didn't work but was not toxic. Maybe the drug worked but was toxic. And there's only really 1 out of the 4 groups in a clinical trial where the drug both had an effect and also did not display toxicity. So if 100,000 people come in, 75,000 of them are just not going to get any benefit from that drug on any given day. DNA is going to change this, or I should say is already changing this, because instead of trying this hit or miss lottery ticket approach for prescribing medications, we are now going to be able to take a patient, take their DNA, study the effects of different drugs on different genes, and say, based on what we already know, you should be taking this medication because your DNA holds the clues and the signals that for you, this is the right medication. So we say that pharmacogenomics is getting genomics in the sense of the right drug to the right patient at the right dose, instead of just trying one drug after the other until we find something that works. Let me show you a few numbers for how this is changing the practice of clinicians and prescriptions. What you see on the left is a graphic that shows you for a few medications, 8 or 10 medications, how many patients after being prescribed in the long term actually have an effect versus how many patients keep taking that medication and are later detected to not have had any benefit at all. I'm going to use the example of Crestor, rosuvastatin. I recently started taking a... Thank you. Yeah, so let's use the example of rosuvastatin or Crestor. I'm sure it has a different brand name here. We now know from long-term studies of Crestor that for every one person that takes or is prescribed Crestor, 19 people will take it, and there will be no appreciable change in LDL cholesterol levels in those other 19 people. So one person will actually see some change, 19 other people will take it and be happy that they are being treated for cholesterol, and they will not realize that this particular medication actually did not have any improvement. Some of them will come back and get a repeat blood test, and then they will be tried on another statin, and if that doesn't work, on a third statin. But mostly the point is that existing ways of prescribing medication that completely ignore genomics are only working for a small fraction of the number of people that are getting them. It's even worse for other things that are Abilify, Nexium. Most patients are not really seeing any differences in their treatment and coming back to the doctor and saying, "Doctor, I still feel bad. Can we try another drug?" So this is what I said about this older paradigm of let's try one medication, let's try the other one, and let's keep trying something until, if we get lucky, we find something that works for you. The new way of doing this is with pharmacogenomics. As I mentioned, in the US, we have about 1,200 medications that are FDA approved. 7% of those 1,200, we know that your DNA holds the clues for whether or not it will work for you. So imagine 7% of drugs already, we could be taking the patient DNA and we could say, I want to prescribe this for you, but I think I'm going to do a genomic test and see if your DNA is saying then this will work for you. If not, I'm not even going to try it. I'm going to start with some other thing, okay? In terms of number of prescriptions, 18% of prescriptions today are written for drugs where DNA information could make a change. This is still the early days of pharmacogenomics. You see that there's more light blue and much less deep blue. But as we learn more and more about DNA and genes and how genes respond to drugs, I strongly predict that in 5 years, this will be turned on its head. And before a doctor prescribes a medicine to a patient, for most drugs, the doctor will be able to say, I know based on the genome that you will have a good response to this drug. And this will result in a much faster effect on these patients who instead would have to come back and just keep trying other drugs one at a time. Okay, in the last two slides or few slides, I want to also tell you about the challenges. I talked about the promises. I talked about how genomics will change medicine. I do want to also be very frank and say that this is not something that will happen in 1 year or 2 years or 5 years. Why? When we have that much information from the DNA, even for someone that studies it all day long, like me, it is very, very hard to understand all of the genetic information from even one person. Computer scientists, artificial intelligence, things are making it easier. But it's not it's not like a classic clinical case where you see the symptoms, you have the differential diagnosis and you say, okay, this is an open and shut case. A genome, to understand it, takes much more time than one of your classical paradigms of treating a patient. Number two, there is a huge lack of diversity. Most genomic medicine studies at this point are still happening in the West with people predominantly of Caucasian ancestry. And you, from your ethics lectures in medical school or from the basic fairness principles of human life, that until you have evidence from your population to prescribe something where the original study was based on a different ethnic group, carries inherent risks of doing harm. I keep using the example of a statin in African Americans, where instead of actually helping heart disease, the same medication that in Caucasians was helping lower cholesterol levels, in African Americans with a specific mutation was turning out to be cardiotoxic. So we really need more diversity in our data in terms of genomic medicine before this starts affecting all groups of society. This is reflected in many ways in the amounts of data. I show you on this slide right now, if we take genomic sequencing, how much of the data comes from different population groups. You can see that European and East Asian, the pink and the blue, are by far taking up most of the space. South Asian, African, Hispanic, these groups are barely visible in the data that doctors have that could guide their patient treatment decisions. So countries across the world are probably going to need to step up. India last week or 2 weeks before announced the release of 10,000 Indian genomes. I think over the next decade, as we have more humans from all countries that were sequenced, this graph will even out and doctors will have more ability to say, I can prescribe this to you based on comparing your genome with other Sri Lankan genomes, not your genome compared with a Norwegian genome, for example. Okay, with that, I'm going to wrap up. I hope I convinced you that genomics matters, that diversity of participants in genomic medicine is a factor. As we get more participants, we need more sequences so that we have things to compare. I'm actually here in Sri Lanka to help train the workforce. Sequencing itself is a fairly trivial act, but until we have enough people trained to be able to process the data, we will be stuck with just the sequence and not a way of translating it for the doctor. So my role here with the embassy and with a local company called Gene Labs is helping create 20, 50, 100 Sri Lankan individuals who have computer science backgrounds and can now work with genomic data. And then I think we will be in a position to say that genomic medicine is now truly ready to take off within the country. Okay, this is my last slide. I know I started with genomics and then I went on to genomic medicine. I think as The Economist, a famous magazine, has put it recently, no other field of science, I think, is touching daily lives as much as genomics. And in the sense, the Human Genome Project, which was 15 years of work, I think has completely revolutionized biology and medicine. We will always have a pre-Human Genome Project world and a post-Human Genome Project world. But I want you to realize whether you're a basic science researcher, a doctor, a nurse, maybe a computer scientist, genomics has fundamentally changed what you can do both for your own work and also to bring the best treatments to your patients at the fastest possible rate we can. So with that, thank you. I'm just going to tell you that the journey to genomic medicine is making progress, but not completely done. That last gap, maybe I will come back and maybe in 5 years, we will see genomic medicine happening. Even in the US, it's not happening everywhere. In Sri Lanka, it's already happening, but I know it can happen at a faster clip. So this journey is more like a marathon, and the embassy and I are here to help you, convince you that we have all of the tools for this, and you are able to practice it in your hospital, in your teaching, in the coming days and weeks. Okay, because there are a lot of young people in the audience, I'm just going to make a quick career plug. It doesn't matter if you're a nurse, an undergraduate, a medical student, a pharmacist, a lab chief, maybe a science writer like my wife. If you have considered a career in genomics, you need to come talk to me. Genomics can mean 20 different things to 20 different people that are studying different things, and each of them could make their own career. I'm a former scientist who is now a money person, right? So my career is a hybrid one, but I know doctors who practice genomics, I know lawyers who practice genomics, I want to leave you with the fact that, especially if you're at the undergraduate level, the possibilities of a career in genomics are endless at this point.